Hi everyone, welcome to today's discussion on gene editing technologies. Today, we'll mainly talk about an exciting, up-and-coming gene editing system and potential source of gene therapy, known as CRISPR-Cas9. To begin, let's look at the history of CRISPR to understand why scientists believe it has so much potential. It all started in 1987 at Osaka University. There, researchers found that E. coli, a species of bacteria, had a strange series of repeating sequences of DNA in its genome. It was not until 2005, though, that scientists realized that there were sequences of viral DNA found in between those repeats, and that this strange set of repeats and spacers, or sequences of viral DNA, could be found in various archaea and bacteria. These regions found in the genomes of these organisms were soon given the name CRISPR, which is an acronym for Clustered, Regularly Interspaced, Short Palindromic Repeats. It is quite a mouthful, but in essence, it's a mechanism that functions similar to our immune system. The way it works is quite complex. However, it can be simplified into three crucial steps. These steps include 1. Spacer acquisition, 2. Production of cRNA, and 3. Interference. In spacer acquisition, special Cas proteins, or CRISPR-associated proteins, capture portions of viral DNA that is inserted into bacteria and incorporate it into spacer regions in their very own genomes. Then, during production of cRNA, bacteria transcribe this DNA into RNA and associate it with a single Cas protein, such as Cas9 or a complex of Cas proteins. It all depends on the species of bacteria. Once this complex is formed, the cRNA serves as a template to allow viral DNA to be identified. This is because the cRNA, since it was directly transcribed from the viral DNA, is complementary to the viral DNA and can therefore hydrogen bond with it. After this occurs, in interference, the single Cas protein or complex of Cas proteins induces a double-stranded break in the DNA and thereby inactivates it. This was a simplified version of how it works, but now let's go into a bit more detail in order to explain how scientists harness the power of CRISPR in order to perform gene editing. First, let's define a few terms. Firstly, cRNA refers to RNA that has been transcribed from viral DNA spacers that are incorporated into bacterial genomes. Secondly, tracer RNA refers to another type of RNA that is transcribed to activate Cas9. Researchers realized that they could make this process more efficient and easier to perform experiments with by linking the two types of RNA to create sgRNA, or simply put, single guide RNA. Not only this, but they figured that if crRNA could be used by Cas9 to identify and cut a certain region of viral DNA, then they could attach any sequence of RNA that they so wished to the crRNA portion of the sgRNA in order to create a certain sequence of DNA. For instance, if they wanted to target a certain gene that they knew the DNA sequence of, then they could attach complementary RNA to the crRNA portion of the sgRNA, and their Cas9 complex would target and make cuts at a certain gene.